Hello everyone. Thank you for listening to this talk. My name is George Calderon de Anda. I've been working at GlaxoSmithKline for about 10 years now, primarily developing batch and continuous crystallization processes for active pharmaceutical ingredients, as well as filtration and drying of these crystals. Today I'm going to present to you the case of a drug substance crystallization where the crystals had a very strong propensity to float in the model liquors. I will cover some manufacturability challenges on scale-up and how we circumvented the particle floating issues. This slide is to provide you some context. So when we do the API particle engineering, we have some information we are given beforehand. So we have some biological targets in terms of what is the target product profile required, what is going to be the tentative dose, and we are also told what is going to be the drug delivery form, for example if it's going to be a tablet or a capsule. We also do material characterization. In particular, for a particular drug small molecule, we screen for the different possible polymorphs that it could have, and primarily what is the most thermodynamically stable crystal structure under which this molecule can form a solid, because the most stable is the one we typically aim to develop. There is also physical stability studies, and there is developability risk assessments alongside. So what is inside this green box of API particle engineering I will describe in the next slide. In essence, we start with molecules in solution. So these have been synthesized through some chemical steps. So that's where we start, and then we design our crystallization, so we form solids in these model liquors. We then filter them and we wash these crystals to remove any residual impurities from the model liquors or any undesired solvent from the process. And finally we dry these crystals. So in the design of these steps, crystallization, isolation and drying, we aim to have certain physical or chemical attributes required for the formulation, such as the attributes mentioned in the bottom of the slide, like particle size, polymorphic form, surface area, residual solvent. So the importance of one or the other will depend on the molecule being manufactured. So for this case that I will present, the solid state, even though we conducted extensive form screens, we could only find one polymorph. So in essence there were no solvates and no other forms, and there was only one anhydrous form. So we didn't really have to worry too much about polymorphic issues. The manufacturing stage consisted of two synthetic reactions and a crystallization in DMSO water. DMSO was the solvent and water was the antisolvent, and these crystals had a high propensity to form agglomerates. These agglomerates were a form of plate-like single particles, as you can see in the picture in the bottom left, and I guess the subject of the talk. So these crystals had a strong tendency to float in the model liquors, as you can see in the picture in the middle, which is the slurry in the funnel before filtration. Even though the true density of the solid was greater than the density of the model liquors, these solids stayed on the top, basically on the surface. This, in terms of manufacturing, this process it poses challenges at large scale, because then we blind to call an endpoint detection for the filtration. The picture on the right hand side is at large scale, is in the pilot plant and the filter dryer, so it's the analogous of the picture in the middle but in the plant. So you can only see from the surface the solids, you can't really see the model liquors which are underneath the solids. And I guess the consequence of this, which I will describe, is when we tried to remove the DMSO from these crystals, it was quite challenging, even though we did a sequence of water washes. So we use, I should mention, we use water to try and remove the DMSO, which is undesired to have DMSO before we go into the drying step because of the high boiling point of the DMSO. So what I will describe here is what were the actual observations when we were processing this compound in the pilot plant. So first you have this little diagram there on the left. So you have your tank, your crystallizer. So that's where you complete your crystallization. So once that crystallization is complete, the slurry is transferred by a pipe effectively to a filter dryer, where you do your filtration, washing and drying. So what I want you to see first is a short video of what we observed in the crystallizer. So in this video you can see is taken from the surface of the crystallizer, so you can see how serious is this floating. And the other observation is you do need to apply certain agitation rate to be able to immerse these solids into the contents. This is important because otherwise you have a particle formation which is different depending on where the crystals are located. And as you can see also there, we're slowing down the agitation rate, and straight away you can see how the crystals float again. So you do need to have certain agitation rate to have a proper mixing. So the slurry from the crystallizer then gets transferred to the filter dryer, and then you end up with a picture like the one on the right hand side, where you have, as I said, pretty much all the solids floating. It's quite impressive to see this. What happens then is you apply your nitrogen for filtration, and then you start charging an intended number of water washes. And after you complete your water washes, samples are taken out of the filter dryer. So samples of this wet solid are taken out, and then it's analyzed by NMR to determine the DMSO content. So here is where we identify the issue, because we had higher levels of DMSO that we were expecting. So we did have to apply extra water washes to then meet the specification in terms of DMSO content. But clearly this was already abnormal. So what we do next is what we did next was conduct root cause analysis. So this is pretty much investigative work where we pull any facts and data of what actually happened during the process. So we have sessions of brainstorm sessions between the people who operated the pilot plant and the R&D people who design the processes in the lab. And then we start to identify the most likely root causes. We do some experiments to bottom up what is the actual root cause. So this is kind of an extract of what happened in that root cause analysis, what came up. But the key question is, why would the particles float in the first place? So there was an extensive discussion here. I just want to highlight three of the kind of lead possible causes. So the number one was, are we entraining gas in this crystallizer by agitation, so that the gas then sticks to the crystals and then form a solid gas structure, which is then lighter than the mother liquors. So this could be by perhaps we're having excessive agitation rate and we are entraining gas from the head space, or perhaps the low fill is low relative to the position of the blades of the impeller, and therefore we have more gas entrained. The second hypothesis was, are we entraining gas during that transfer from the crystallizer to the filter dryer? Because we use nitrogen to push that slurry all the way to the filter dryer. And the third hypothesis was about the particles themselves. So are we in some way modifying the process so that the crystals actually grow with different physical properties, and perhaps some of these properties lead to more propensity to float. So the consequence of that is if we then end up with solids floating in the filter dryer, so when we charge the washes, we would then have poor penetration of the wash because the water may be bypassing the cake, the solids. They don't really penetrate between the gas and the particles, and therefore they don't really remove the DMSO. Perhaps there may be channels through which the water prefers to go rather than washing properly the crystals, and therefore giving us the reason why we observe high levels of DMSO. So as part of our investigations, our molecular modeling group looked at what would be the predicted morphology of these crystals based on the crystal structure. So because you know the crystal structure, you can then associate which functional groups are exposed or are associated to each of these phases, and from there derive what is the relative wettability in terms of which phases are more hydrophobic or more hydrophilic. So the main outcome of this modeling was actually that the largest phase in these crystals, which is the 001, was actually the most hydrophobic. This was already a hint because we are operating in a system which is aqueous. So really, with these large hydrophobic surfaces, it means that any gas bubble that gets entrained will have a propensity to interact with these crystals. In terms of the actual experiments we conducted, we built this mechanistic 3 factorial study to look at what is the effect of the 3 main mechanisms in that crystallization on the physical properties of the crystals, and as well as on the degree of floating of crystals after the process is complete. So we did look at seed dispersion. We found a way to favorably disperse those seeds and unfavorably, say, a poor dispersion of seeds. Mixing, which is simply increase or decrease the RPMs in the experiment. So the high RPM corresponded to a scale down agitation rate from the pilot plant, and a low RPM was just enough to suspend the solids. And we also have some anti-solvent addition, which is a combination of DMSO and water. So it's whether we add fast or slow this anti-solvent, we would then generate high or low subsaturation. So the hypothesis was that maybe the high subsaturation helps to fuse more those particles or those agglomerates, and therefore would lead us to a different set of physical properties of those crystals. So we could lay out these 8 experiments. However, we didn't actually have to execute all of them. By the time we ran about half of them, we already knew what was going on. So the setup was a 1 liter scale. It's a geometric mimic of what the crystallizer is in the pilot plant, and we had our PBM probes and our FBM probes in the vessel, and we also measured some concentration and levels of DMSO offline. So this is a set of the PBM images we got when we were executing the experiment at high agitation rate. So these images actually are quite telling. The first one on the top left, which is just moments after we charge the seeds, even though we use the favorable seed dispersion conditions, we observe that actually the seeds don't really disperse, they remain as clusters. So these seeds are actually dried meal particles of about 5 microns each. But when we charge them, they tend to stick together, and then as they grow, no wonder that you end up with agglomerates, so you basically grow in those clusters. But probably more relevant to the floating aspect of the process, we very clearly saw from PVM images that there were some droplets of gas being entrained, and it was very clear how these bubbles were actually interacting with the solid, and became it was apparent that this gas was forming these structures of gas solid that we were talking about before, which then lead us to what we say as foam, which is basically gas and solid. The FBRM data again was also telling us that the greatest impact came actually from agitation rate. So if you see the chart on the top with the total counts, so the two lines on the top correspond to the experiments at increased revolutions per minute, and the two on the bottom lower revolutions per minute. So this is where we see the greatest change in total counts. So you see that even though we change the way we disperse the seeds, actually the counts lead us to very close to each other, and it is not really variability in the experiment in terms of the seed dispersion. And similarly for the anti-solvent addition, whether it goes fast or slow. And again, the distributions in the bottom is just obviously it's an approximation of our size, but then again, the particle, the physical properties don't really change much. There is a slight difference in physical properties, as we confirm with other techniques as well, when we had low or high agitation rates, but not really when we vary the seed dispersion or the anti-solvent addition rate. And of course we had the PVM images, which were also consistent. The observations were consistent that there was more gas entrained when we had high agitation rates. So the conclusion, I guess, from these in situ techniques was the agitation rate has the greatest impact on what is happening in the process. And then I will show you next what visually we see in this crystallizer. So as you can see the pictures on the top left, where we have a low or high agitation rate. So this is when the process is finished, you stop the impeller, and after about 5 minutes you see how different is the quantity of material that is floating. So it's a much thicker layer of crystals floating, or foam really, when you use a high RPM. So before I describe the rest, I will show you another short video, which is a PVM sequence of this moment when we stop the impeller. So in this video you see the crystals here being stirred, so this is the end of the crystallization, and then all of a sudden we stop the impeller, and you can see how immediately these bubbles of air start kind of pairing with the crystals, with these agglomerates, and these are being dragged up to the surface. So now this really is very powerful experimental evidence of the mechanism of floating. So it's not just the modeling now, it's actually we're actually getting hard data to demonstrate what is actually happening with this floating. So now we figured out then, okay, so if we entrain more gas, then we can basically increase the quantity of material floating. So if we then were going to entrain more gas artificially, what would the result be? So what we did was then we offload the slurries from these two experiments, and then we shake the bottle, basically the shot bottle where we put the slurry. So we artificially entrain more gas, And what we found is we can actually make most or all of the solid, pretty much virtually all the solid, to float, as you can see on the right hand side. So this is after it's been shaken in the bottle, then you pour that slurry into your glass funnel. You can see how all the solid is floating. So even though we use low RPM in our crystallization, by that artificial entraining of gas, we can actually make all the solid to float. So this is relevant because, as I said, there is a transfer process from the crystallizer to the plant where you use nitrogen, and it is not a trivial step, because we do have to use some nitrogen, as I said, and this would entrain more gas into the contents of the slurry. And I guess the only other point to make is there was really minor impact on particle size distribution and agglomeration of the other mechanisms studied, which were the seed dispersion and the anti-solvent addition. So here I just want to explain to you what we would normally expect of a filtration and washing process. So the little drawings on the top left really, mind you have your filter there, you charge your slurry first. So what we normally do first is to leave the slurry settle basically for a while. So we leave for good 30 minutes in the pilot plant for the solids to form some bed and the bottom of the slurry, and then you will start to see the mother liquors getting clear on the top. So after this settling period, we apply a driving force, vacuum, or typically in the pilot plant is nitrogen, to then start the filtration. So what happens then is the liquid starts to go through the bed of solids, and when the surface of the liquid hits the surface of the solid, we say this is the dry land point, so we stop the filtration at that point. And then we follow with the wash, which is basically you charge your wash on top of that wet solid, and then again you push the wash through, and in theory you would need about 1 cake volume of wash to displace all the mother liquors. However, in practice you normally need more than 1 cake volume. So this is how you would normally do a displacement washing. Now in our case, obviously we had this floating of solids, so even though we leave it to settle for a while, they don't actually settle. So even if you leave it overnight, it's a very stable structure which stays floating pretty much. So if you imagine applying vacuum to that slurry, as you see, so the filtration will go very fast, because you then start passing the clear liquor first, and then at some point that solid will go to the bottom. So the concept of dry land here, it doesn't exist. So all we can do is to deliquor all that solid, and because there's no way to call the dry land point, so we just finish the filtration where we don't see any more liquor coming through in the bottom. So when what we saw is when we charge the washes, the water washes, we either saw total refloating of solids or partial. So this was clearly indicating that there is still gas in between those crystals, and this means that actually the water wash is not actually breaking through the gas and the solids, so it's not actually washing. And you could also see this visually when you charge the wash and you apply the vacuum. You could see how the water was either bypassing the cake or finding its way through some holes. And the other observation was after a number of washes, you could see that at some point the cake started to be degassed, and then the solids stopped to float. However, even at lab scale, this was not consistent, so there was not a particular number of washes where you can say, yes, at this point my solids will not float anymore. It was very difficult, and all this refloating problem really gives a very inconsistent behavior of the cake. And so this filtration basically is very difficult to scale up, in essence. So if you cannot reproduce it in the lab, it's very hard to believe that you will be able to run it consistently in the plant. So going back to what's happening in the pilot plant, so we said there is a transfer line which goes from the bottom of the crystallizer to the filter dryer. So in a diagram it's just an arrow, as you can see, but in practice is the photograph on the right hand side. So you actually have your crystallizer in the top floor and your filter dryer in the floor underneath. And when I was there, I counted that pipe had at least 21 bends plus all these flanges here. And I want you to see a short video again. So this transfer line here below all these flanges has a side glass, so you can see the slurry passing through. So I will show you a video of this slurry coming through. So this is the side glass, and this is the slurry coming through. As I said, it's being pushed with nitrogen. So as you can see, even though we use low pressures of nitrogen, that stream is very turbulent. And this is the filter dryer, so this is the slurry coming through. So there is like a deflector as the stream comes through, so just to avoid the stream to jet directly down into the filter dryer. So you can see it's not a trivial step of just transferring. So even though you can control your agitation rate in the crystallizer and minimize the floating, clearly you will have gas entrained during this transfer to the filter dryer. So I guess the message I want to give you is no wonder that you would end up with the solids really floating in that filter dryer, no matter how careful you are with your crystallization in terms of how much gas you get entrained in the contents. By having this turbulence and more nitrogen entrained in the slurry, you basically cause some severe floating, which would then lead you to the observed issues of washing DMSO. We did investigate other methods to wash this slurry. So there is a method called Re-slurry washing, which is basically you charge the water into your filter dryer, and then you use the impeller in the filter dryer to stir those crystals in the water. However, this impeller is not as good as in a crystallizer, because it's not designed really to stir slurries, it's designed to stir powders. So what we found is it was very difficult to implement because the mass of solids is floating, so you do need to move the impeller up or down to the zones where the solids are. And in essence, the solids and the gas don't really get stirred properly, they just move along with the impeller but don't really get a proper mixing. And the values of DMSO that we were measuring by NMR are pretty much comparable to the displacement washes. So all this experimental evidence was telling us actually there is no benefit in doing Re-slurry washing over displacement. So that led us to looking for an alternative solvent wash. And in case you wonder why you didn't do this from the beginning, that's to do sometimes with the status of the project and what things you are flexible or you will not to modify. But in essence, we set out a series of success criteria, and clearly one of them was the particles should not float in the new solvent wash. And there are other criteria that's to do with delivering the purity of core, removing the DMSO, and make sure you are not going to be dissolving your crystals significantly. So we identified some candidate solvents. We selected in the NIPA water, which was the most promising. So the pictures here are first test we did at 60 milliliter scale in filter tubes. So the picture on the left is the slurry, with the model echo. So you see all the crystals are actually floating. The picture in the middle is once you charge your DMSO water wash first. So you see how the crystals float as we saw before. And the picture on the right hand side is with the new solvent wash, the IPA water. I'm not sure if you can see very clearly, but all the solids are actually in the bottom, and you only have clear liquors on the top, so that's quite encouraging. So we were pleased with this result, so we decided to scale it up to 1 liter. So we have in the lab a 1 liter filter dryer, which is a mimic of the one in the pilot plant. And the picture on the top left is once you charge that IPA water over your solids, you can probably more or less see that most of the solids are actually in the bottom. There are a few solids in the surface, but it's really not a significant quantity. So again, this was quite encouraging. And the values of DMSO removed over the different washes was again telling us we're actually removing the DMSO this time. So by the time we did 3 washes of IPA water, we pretty much had low enough levels of DMSO to call it a success. And the pictures in the bottom is just a confirmation that we don't have an adverse effect on the physical properties of the crystals once we dry them, because we now introducing a different solvent system. So all these seem to be like a very encouraging method to circumvent the issues of floating crystals. Unfortunately, this project was terminated before we could run it again in the pilot plant, so we didn't actually have the opportunity to run this new method at plant scale, but we believe it would have been a success, basically, based on all this laboratory scale data. However, the question was still, why would the IPA water prevent floating compared to the DMSO water? So we look back at the values calculated from molecular modeling in terms of wettability, and we found that the values calculated for the wetting with IPA water and DMSO water were comparable between each other. But when we compare these values to the surface tensions of the solvents, we did notice that the wetting values calculated are significantly lower compared to the surface tensions of water and DMSO, but are pretty similar to or close to the surface tension with IPA water. So we think that this may help explain perhaps why there is a better penetration of IPA water in between the crystals compared to the penetration observed, the poor penetration observed with water. We didn't really get to the detail of this because of the status of the project. It was terminated, so there's really not much effort put on understanding the details, but we think that the explanation may go with the surface tension and the wetting energies calculated. So just to conclude, I guess some of the key learnings. Clearly, in pharmaceutical crystallization design, we require techniques which are sensitive enough to the various mechanisms that are involved in our processes. For this particular case, I presented the mechanisms or the key mechanisms were clearly seed behavior, crystal growth, agglomeration, and facet surface chemistry. So any technique really that can be sensitive to these mechanisms is gonna really help us. In this particular case, we thought imaging was quite powerful. There is a lot of information contained in these images. For example, if you consider the image here that I put on the bottom, if you look at it, you can tell that there are agglomerates. You can tell what is the particle shape and size of the single crystals forming these agglomerates. You can tell there are bubbles. You can tell the bubbles are interacting with the solid. You can tell how many crystals are there, more or less, how much material has come out of solution. So all this information is contained in just one image. So imagine we producing, basically the instrument is producing hundreds of images. So as a user, I will not be able to look at every single image. there may be trends over time in this image. So how could we unravel these trends? How could we have perhaps a way to unravel information such as what would be the percentage of agglomerates versus single particles in this system? Is the shape of this agglomerate changing over time? So that's information that is very difficult to see by eye, especially over time. So in GSK we're really interested in having techniques that are able to somehow extract quantitative information from these images. We've actually spent quite a bit of effort in the past years looking at imaging. In the past we looked at systems at line, like flow cell system, where we took a sample of line and we could analyze it straight away, but it's offline nevertheless. We also look at setting up cameras, looking through the wall, through the jacket of the crystallizers. And we most recently acquired obviously PVM instruments, which are able to image from the beginning to the end of our processes. And we've set up collaborations with Strathclyde University looking at possibilities to analyze these PVM images. We also looked at some algorithms developed in-house with MATLAB and commercial software, trying to extract quantitative information from the PVM images. So it's always work in progress. There are pros and cons. I don't think there are techniques still able to analyze a full sequence of crystallization process images. However, we think there is a lot to gain from these images. If we could possibly extract quantitative information, I think it would be a significant step forward in understanding our processes. So I would like to just acknowledge finally Francesca Percivali was a PhD student at CIMAC who conducted some of the experiments I presented here, and obviously our modeling group at GSK for conducting the molecular modeling that I described in this presentation. And I would just like to thank you very much for your attention, and I hope you find this interesting and useful.